Okay, so first of all, I would say it's a very great pleasure to have this opportunity to talk to you all. I very much regret that I'm not doing it in person. I did set out on the journey. I actually got pretty close. I got to Calgary, but I couldn't make that final part across to Lethbridge. But hopefully the Skype will give me the opportunity to tell you about one of our exciting programs here at Rothamsted. Because I'm doing this by Skype and because I won't have the opportunity to talk to you over the coffee breaks or in the bar, then I'm going to make sure I leave a decent chunk of time at the end for any questions you may have. As you will know, I'm from Rothamsted Research in the UK. Rothamsted is the oldest continuously running research centre in the world. It's been running for more than 170 years now, and we're spread over 3 sites. If you look at the map of the UK on the right-hand side of the picture there, you can see our main site in Harpenden, which is just north of London. Then we have a site in the West Country called Northwick. The site there is focused very much on grasslands and farming systems. And what they've divided that farm into is 3 small farmlets, where they're measuring all the inputs going into it and all the outputs from it. And this allows them to use a systems approach to see how changing farming practice affects the numbers for the inputs and the outputs. And on the right-hand side of the map, you can see we've got our site at Brooms Barn, and that is focused on sugar beet research. Overall, within the Institute, we have around 300 scientists at any one time, and they're well supported by technical and greenhouse staff, etc. And we have 50 to 100 PhD students. And just to give you an idea of the numbers, our annual budget is around £30 million per annum. So although we have these 3 sites, we actually share a single vision, and that's to be a world-leading bioscience research centre in plant and soil science for sustainable agriculture. Now, we believe that we live up to our past. Many of you may know that the Institute was founded by Sir John Laws, and he was the guy who invented the first inorganic fertilisers, which have allowed the development of most of our agricultural system. You can see that as well as an interest in chemistry and nutrition, at Rothamsted we have a long-standing interest in statistics and biomathematics. And the work at Rothamsted was the basis of all our modern biological statistics. Fisher at Rothamsted developed the use of the Latin squared, the F-test, the correlation coefficient, and analysis of variance. These are things that we're seeing and using in our everyday work. Now, another major discovery from Rothamsted was the discovery of phenoxy acid herbicides. Of course, the most well-known of these is 2,4-D. This is the most widely used herbicide in the world. It was particularly important as it was the first selective herbicide we had, killing off the dicots and leaving our monocot crops. More recently, at Rothamsted, we discovered the nitrogen assimilation pathway in plants, which involved GS and GOGAT. And this, as many of you may know, is a target for herbicides like Liberty Link. Now, another major achievement at Rothamsted was the first synthesis of synthetic pyrethroids. Now, these are widely used in pest control. They have a low mammalian toxicity and a high toxicity against the target organisms. So on a worldwide basis, they now account for over a quarter of the pest control agents that are used. Unfortunately, we don't benefit a lot from that at Rothamsted, as we hadn't got the royalty position sewn up at that stage. Although our experiment and our station was founded 170 years ago, some of the first experiments that were put in place then are still running, and over that time we've maintained strong samples and archive material. Now, this has been very useful to us because it allows us, for example, to follow the development of diseases how strains have changed over time, and even, for example, to follow things like the nuclear tests. Now, like any organisation, we have a mission statement, and ours is to perform world-class research to deliver knowledge, innovation and new practices to increase crop productivity and quality, and to develop environmentally sustainable solutions for agriculture. Now, the way that we're doing that at Rothamsted is within these 4 Institute strategic programs. You're going to hear a lot from me about 2020 Wheat, but I'm just going to briefly mention the other ones that we have. They're all quite closely related. Of course, it's important that whatever we do in programs like 2020 Wheat is sustainable. Therefore, we have a program which is focused on sustainability. That's particularly focused on designing, modelling and testing novel agricultural systems, and putting a price on things like ecosystem services. We have another area which is focused on cropping carbon. That is focused very much on trying to reduce the agricultural footprint, and there the main focus is on using short rotation coppice willow as a bioenergy crop, and at the same stage trying to look at agricultural systems and the way that we can lock carbon up into the soil. And then the final one that I haven't spoken about yet is designing seeds. And what we want to be able to do is design the seeds that they have the appropriate quality for whatever they're going to be used for. But our particular focus is on diet and health. So we have work there on canola and on safflower, and we also have work on wheat. And obviously the work on wheat aligns closely with the 2020 Wheat programme. Now, of course, everyone is aware of the perfect storm that is brewing. We know that there's going to be a huge increase in demand for food. We know it's going to need to increase by around 50% by 2030 and double by 2050. If you go to different sources, the numbers vary, but they all show the same trend. We know that demand is driven by both the rise in population. So we know that we're going to have to meet this challenge with less in the way of resources. We've got less land. And also, we know that a lot of the things that we do in agriculture exacerbate problems like global climate change. Probably the best example is in terms of the production of nitrogen that we use to fertilise our crops. So we know that we're going to have to use those in a more efficient way and at the same stage increase yield. What I'm particularly interested in is wheat. We know it's one of the staple crops in the world, 20% of the global daily protein and calories. We know that we need to increase the yield. Wheat is grown on a wide geographic range, and in the UK it is our crop. Now, if we look at the wheat yields around the globe, you can see in Canada they're fairly modest. In the UK, we're talking around more than 8 tonnes per hectare. And there's a lot of different reasons for this in terms of the types of wheat that are grown in the different areas. So in the UK, the wheat is grown mainly in the east of the country. 95% of what we grow is winter wheat. It's in the ground for a long time. Typically, the crop will be sown in September and harvested in August. So it's in the ground for 11 months. The average yield in the UK is 8 tonnes per hectare, and we achieve that using around 180 kilograms of nitrogen per hectare. And of course, the market value of the crop and the food value of the products that come from it is very high. Now, if we look at what's been happening to yields in the UK in recent years, you can see that they went up very rapidly in the 60s and early 70s, but in the last decade, they've plateaued, and that is a real concern for us. So 20 years ago, the average yield in the UK was 7 tonnes. The world record is at 15.6 tonnes. In the UK, our record is a little bit lower. It's about 14.3. And Rothamsted, we've now got our 2020 wheat programme, which in the next 20 years is aiming to increase the yield of wheat to 20 tonnes per hectare. And on the right-hand side, I've just got a picture from some yield mapping that was generated by one of our trustees. And you can see that there's large variation in the yields that we get over a field. And our challenge, of course, is to try and make that yield as uniform as possible. Now, can we learn from history? This is our founding father, Sir John Bennett Laws. He established some of those early experiments that I was talking about. There's one here which is a particularly classic one called Broadwalk. It's a nutrition experiment. It was put in the ground before we'd had that work on statistics, so it's not replicated. And what we have here is a single of strips going up the field with different levels of nutrition. And the bands across the field are where there's changes in farming practice. You can see that some of the results from that. And you can see that over the course of the experiment, but particularly in the last 30 or 40 years, there have been big increases in yield. And this has come as a result of a combination of factors. There's no single magic bullet that's delivered it. It's improvements in agricultural practice, the introduction of crop rotations, the introduction of herbicides, fungicides, and of course also the genetic improvement of the plant material. So our project 2020 Wheat aims to provide the knowledge base and tools to increase the potential yields of wheat to 20 tonnes per hectare within the next 20 years. And of course this is for the UK. Now, to deliver a program like this, we need a long-term strategic program, and we need to bring together a range of disciplines in an integrated approach. And we see there being three major challenges that we face. One of those is to increase the yield potential whilst maintaining quality. Now, quality is always a difficult thing because it depends who is going to use the wheat at the end of the day, and the requirements are different, but we have to be mindful of quality. We want to minimise the gap between the yield potential and a farmer's actual yields. And we also, if we need and want what we do to be sustainable, we need to maximise the efficiency of input use. So within the 2020 Wheat project, we have a number of clusters. One of these is called Maximising Yield Potential, and it does what it says on the label. It's got work inside it that's focused very much on increasing the photosynthetic capacity of the canopy, using nitrogen efficiency, making sure the architecture is right, and we have the appropriate fertility. Another of the work packages is focused on predicting yield potential, and there our focus is on just two particular pathogens. There are, of course, other things that we could target within that package, but we have limited resources, therefore we've chosen to focus on those which are most important for the UK at the moment. We then have a work package on soil-resource interactions. There we're looking at the structure, the root function, the root architecture, and also another soil pathogen, one which causes the second wheat syndrome, or otherwise known as takeall. And then tying it all together, we have a systems approach to crop improvement and modelling. Okay, so what traits are we interested in? This slide shows some of them. We're interested in biomass. We're interested in yield and yield stability. We're interested in the fertility of the plants. That's in relation to number of grains that are set, the size of the grains. We're interested in photosynthesis. We're interested in carbon partitioning. What determines harvest index and can we make it better? We're interested in resource use efficiency, particularly water, nitrogen and phosphorus. As I've said already, we're interested in a number of pathogens. Of course, we're also interested in other traits related to quality and pests, but at the moment, those don't fall within our programme, although there's some other work in the Institute on these, which is funded by other sources. So what we have is really two basic hypotheses. One is that there's existing variation out there in germplasm that we can exploit, and the other is that in some cases we can manipulate key traits using genetic manipulation and get the benefits in that way. So what kind of variation are we looking at? We're looking particularly at diversity panels, and there's some examples there. The Watkins collection is an interesting one. It was collected in the 1930s by British diplomats who sent back seed from the local markets. That was put into a collection of many thousands of accessions and is really exploring some of the widest diversity there is out there. There are also mapping populations which have been produced, which allow us to dissect some of the causes of genetic variation that we see. There are the newly created synthetics, which are produced by our colleagues at the National Institute of Botany in Cambridge. We've also been looking at variation that's come from the ancestral introgression. That's done primarily with some of our colleagues at Nottingham. We've also used mutagenesis, and of course, we also generate transgenic plants. We also have a number of technology platforms that enable us to do this work. We have a strong wheat transformation group, and I'll say a little bit more about that in a minute. But we've also used mutagenesis. We have saturated populations of both hexaploid and tetraploid wheat. And we can use these both in forward screens where we're screening for a specific trait. And some of the traits that we've been looking at are yield stability, and pest resistance. But we can also use them in a reverse genetic way to identify mutations in any specific gene. A new technique that we've recently developed is viral-induced gene silencing. It allows us to give transient gene silencing and protein expression throughout the wheat plants. Everybody's heard of the omic technologies, and we're exploring those. And then we come to two other areas which are really important to us. One is bioinformatics. We're doing a lot of experiments with diverse germplasm, so therefore we need to be able to store the data that we get from those in a way that we can interrogate it. We also need to be able to have small programs that enable us to do the modelling of crop development. And we also need programs that enable us to use the genomic information that becomes available to us. And then, of course, if we're looking at all this germplasm, another area that's important to us is being able to accurately report on the traits that we're interested in. And that we call phenotyping, and need to do a lot of it. Now, clearly, another thing that we're interested in is trying to exploit that vast amount of information that's come out of it from sequencing the wheat genome. We know that if we have that, it'll make it much easier for breeders because we'll have perfect markers for many of the traits. It'll also be able to help us make some of the reverse genetic approaches, such as tilling and talling, more efficient. And it will also give us new tools that we can use in the generation of, for example, transgenic plants. So this is just a picture of one of our fields at Rothamsted. In this, we have more than 11,000 plots, and for each of those plots, we need to obviously get as much phenotypic data as possible. So what we want to be able to do is, by describing that germplasm, is be able to predict how it will perform in a given environment. And to get to a stage where we're able to do this, what we need to do is, first of all, have the germplasm. And I think I've already suggested to you that there's a lot of diversity out there that we can look at and examine. We need to know about the genes and the sequences. And although we haven't yet got the complete sequence for the wheat genome that's fully assembled, we're coming close to that. And I don't believe that that will be a bottleneck. But where we have bottlenecks at the moment are in relation to describing the performance of those plants in the field and being able to store and manipulate the data on them. So if we're doing phenotyping, then what we've been doing in the past is using a series of handheld devices which are looking either at the crop or the environment, or perhaps the seeds once they've come into the lab. But what we want to move to is a more automated system. And here you can see some of the toys that we're interested in. In the centre there you can see an octocopter. So clearly we can see differences in temperature which we can use to measure plant performance, particularly in relation to the use of water. And then you've got the kind of gantry system at the top, which is something that we'll be installing at Rothamsted within the next few months. So one of the other things that we're trying to do is take information that we might get from other crop species, put them into wheat plants. And what, of course, we're interested in is not just the performance of individual wheat plants, but the performance of whole canopies. So one thing we have at Rothamsted is a good wheat transformation group. This is the largest public sector wheat transformation group in Europe. It has more than 600 unique events each year. We can do the transformation using agrobacterium or biolistic methods. We can transform any genotype that we've looked at least 40 elite genotypes already. This is important because if you're trying to make a plant better, you actually want to start off with a good plant rather than something which is only suited to grow well in a researcher's lab. And also to target the gene expression, you need the appropriate promoters, and we've been validating those. And then, of course, in the UK, we don't have a large experience of GM crops, but we do have experience at Rothamsted of GM field trials. So one of our targets, as I told you, is maximising yield potential. Then we know, what determines crop yield. It's biomass times the harvest index. And the first green revolution was really built around introducing dwarfing genes to our crops. They had two effects. They increased the harvest index, but they also allowed us to use more nitrogen. Okay, so allowed us to use more nitrogen. But that harvest index is already quite high, it's 0.6, and therefore if we want to substantially increase yields, we're unlikely to do this because we're always going to need some infrastructure to hold the ear and the grain. So really we need to increase biomass. And if we look at where biomass comes from, it's a function of the light that's intercepted multiplied by the resource use efficiency. And if we look at the light interception by our crops, again, that's already high. So this tells us that if we want to increase the biomass, then we need to increase the resource use efficiency. And if that's what the next green revolution is going to be about, that's what we're going to have to do. And that means we're really going to need to increase photosynthesis. If we look at the energy that comes from the sun and how little of it is actually used productively in photosynthesis, then you can see that there's a process where there appears to be substantial room for improvement. And the most noticeable thing that you see there is the difference between C3 and C4 crops. C4 crops are like maize, C3 crops like wheat and rice. And it's increased by about 50% if you go to a C4 plant. And this is because their primary CO2 fixing enzyme, which is called rubisco, has a pump to increase the CO2 and therefore increases its efficiency. So that's one way in which the resource use efficiency of the maize is higher. And therefore it's not surprising that we know that increasing photosynthesis works. A large number of experiments have been done where they bled CO2 from pipes into the field and looked at the performance on crop plants. Here is a summary of a big analysis that was done with more than 400 experiments looking at 37 species. They found an average increase of around 30% by doubling the CO2 concentration. Now, if we look at the underlying biochemistry, it looks complex, but it isn't that complex. And from it, we can get a number of clues of areas that we could target. Right in the center there is the enzyme that I talked about, rubisco, that plucks the CO2 from the air and fixes it into sugars, which yield to yield. Now, that enzyme as well has a wasteful reaction with oxygen, which isn't very helpful. One way you can overcome that is by pumping the CO2, and that's why the CO2 enrichment works. We can try and improve this by, as I said before, looking for natural variation or by targeting specific steps, perhaps using GM interventions. Now, if we look at the biochemistry, we can see that within that process, there are two limiting steps. The first ones are limited at the low CO2 concentrations by the characteristics of the rubisco itself. That's the blue dotted line. And then higher up at the elevated CO2 concentrations by other characteristics within the leaf, shown by the pink line. Now, where a plant sits at any one moment in time is going to depend on other environmental factors. So again, looking at the biochemistry and looking at that RUBP regeneration phase, the pink one, we can identify steps where we believe from models we could increase performance if we increase the amounts of some of the enzymes in that cycle using a GM or a selection approach. Why do we think this? Well, we know it works in tobacco. These are tobaccos in which just one of those enzymes has been increased, and I think it's quite clear to see that the yield and the biomass is much greater in the transgenic plants on the right-hand side. So, will it work in wheat? That's what we're trying to establish. We've used our wheat transformation system. We've got small plants. When we introduce a gene like that, we couple it with herbicide resistance. That allows us to select for it. We then get the plants and we can start to screen them. So here we have our tool, which is infrared gas analyzers, which measuring the photosynthetic performance. And the next click of the button should put a little graph up on the right-hand side. And we can see that some of our transgenics appear to give a small increase in that RUBP regenerated phase of photosynthesis. And even small increases like this should lead to fairly substantial increases in yield. As I said before, we know that we can bleed CO2 into the field to increase the CO2 concentration, but we can look at other approaches. We can look, for example, at the frequency of stomata. We can look at the diffusion of CO2 through the membranes. We could try and introduce the Cran's anatomy that you find in maize to wheat plants, or we could try to learn from other organisms like the cyanobacteria and the hornworts, which have other methods for concentrating CO2. One that we're trying to use is exploit the CO2 concentrating mechanism that is found in cyanobacteria. This is a joint project, as you can see, with California, Berkeley, Cornell University, University of Illinois, and ourselves. And this is trying to use synthetic biology to put in a carboxysome to concentrate CO2. And the carboxysome is that little blue shell-like structure you can see on the right-hand side. So the important thing about these carboxysomes is that they have a protein shell, and therefore we can try and recreate that in our crop plant. And the way to do that is to introduce all the building blocks. And the key things that we're trying to do at the moment is define exactly how many of the building blocks we need and can we get them to assemble into something that which will function. And the answer is we've managed to introduce a number of them, and we are getting them to assemble into punctate loci, those spots of fluorescence that you can see on the images on the right-hand side. And if we look at even higher magnification, we can almost start to believe that we're assembling objects with that kind of shape that we'd expect with the flat sides of the carboxysome. Of course, we're also very interested in nitrogen. We know that on the downside, the manufacture of nitrogen has an important carbon footprint. We also know that nitrogen can leach from the soil and damage water courses. But of course, on the plus side, we know it enables us to achieve higher yields, and it's also very important in determining the quality. The normal nitrogen response curve we might expect is illustrated by that dotted line in red, and the scenarios that we'd like to achieve are shown by the dotted line in blue and the solid line in blue. So what we want to get is more bangs for our buck. We want more yield with less nitrogen. Now, this is just giving an example of some of the germplasm screening that we've been doing, looking for variation in those nitrogen responses. This is in relation to grain yield and by comparing plants that are growing at two different nitrogen levels. So when we're interested in a trait like nitrogen, then you can start to break it up. We're not just interested in nitrogen use per se, but we can think of it as things that are important, are the amount that's taken up from the soil, the amount that's utilised by the plant, the amount that ends up in the grain. And when we're looking at those processes, then we can start to look at the plant traits that are important. So in terms of the uptake, then how quickly the roots become established, their distribution through the soil and their ability to abstract nutrients and water together are going to be important. If we look at the nitrogen utilisation, then we're interested in how quickly we can establish the canopy processes like photosynthesis, which I've talked about before. And then when we think of the nitrogen harvest index, then things that come into play are like the rate at which nitrogen can be transported from the leaves into the developing grain to give it its quality. So this is again just showing some of the variation in those specific traits in a range of wheat cultivars. And you can see that what you'd ideally want is a pink band right the way across all of those traits: yield, utilization, harvest index. But you don't find that. And this allows us to start to work out why cultivars do or don't perform well as far as nitrogen use is concerned. And this is just a reminder that we can then go from that to look at, for example, changes in gene expression or the levels of metabolites to identify specific gene targets that we can either manipulate directly or test in mutants. So, as I've said, we're also interested in the architecture and fertility. We know that the last green revolution was built around that, but we also know that the variation that is currently exploited in our wheats is not optimal because there are a number of pleiotropic effects as well as the shortening of the straw, which can influence fertility and can influence stress tolerance. What we'd really like is a specific target that is only going to target the stem extension. If you look at the metabolic pathway on the right-hand side and the gene list, you can see that the RHT is as a single gene that has these pleiotropic effects and is expressed throughout the plant. But some of these other steps are unique and only expressed by multiple genes in different tissues at different times. And therefore, by targeting these, we can directly manipulate height. So one thing that we've done is use our mutant populations for this. And if we can have one more click, then you can see that in each of the three genomes that we have in wheat, the A, B, and the D genome, we've identified mutants. And we've been able to combine these and introduced a new wheat which has a shorter straw specifically. So in terms of protecting yield potential, we're interested in these two diseases, Septoria and Fusarium. Fusarium's, of course, important because it not only affects the amount of grain we get, but also the quality, and that, of course, also has implications for human and animal health. So our basic approach here has been to know our enemy. We're particularly interested in the pathogens themselves. For the Septoria, it normally has 21 chromosomes. These have been sequenced and analysed in detail, but what we've discovered is that 8 of these chromosomes are dispensable. So if you've got a control strategy, it's no good targeting anything that's on these chromosomes. It has to be on those core chromosomes. And also, by analysing the information there, we can identify new targets for intervention. And here we're looking particularly at those proteins which are involved in the secretome. They're the proteins that are secreted and have their first interaction in the apoplast of the plant. So what's important is if you can stop and prevent infection at that early stage, then you'll be able to minimise the damage. So we're also interested in soil resource interactions. And one of the main traits that we're interested in, water. Here you can just see some data that we've got on the right-hand side in a collaboration with the University of Lancaster. Using electrodes, we're able to monitor the water in the soil continuously and be able to generate these 3D models of water availability. You can see the little red patches underneath the green blocks show the amounts of water that are being abstracted from under the different crops. And this gives us an indirect measurement of rooting performance. Another way we can try and get similar sorts of data is by the ability to press a probe into the soil and measuring the resistance that has, which is a factor of soil strength, which again tightly correlates with water availability. So we have a number of other techniques that we can use in the growth rooms where we can look at the root architecture in paper rolls, we can look at root performance in hydroponics, and also we can separate out the effects of strong soil and water availability by growing plants in individual columns. So the pathogen that we're interested in here is that second wheat syndrome take-all. It causes these nasty patches. It's very important in the UK, a major constraint on our productivity. If we go to the next slide, you can see we've been looking in one of those collections I talked about at the build-up of take-all in the soil and susceptibility to take-all, and you can see that there's substantial genetic variation that we are now exploiting. So the final work package that I want to talk about is modelling idiotypes. And this is really important because it allows us to test on the computer what combinations of traits will be advantageous as far as yield's concerned, not only for today, but also for future environments. So what we're able to do is take our model of wheat performance, link that into data that we're getting on climate change and weather generation, and design ideal idiotypes for future climates. So you can see here that we've done some analysis, and we know that for the future, the climate in the UK is going to get drier in the summer. Now, most of us are quite happy about this, but we had thought that it was going to create problems with drought and wheat yields. But in fact, the model predicts that that's going to become less of a problem in the future. And this is because most of our crops will escape that dry weather by maturing earlier because of the elevated temperature. However, in contrast, those elevated temperatures are going to be more concern. Now, this is important for someone like me because should I be investing my money in drought research, or should I be investing it in ability to cope with thermal shocks, particularly around the time of flowering? So we can also put these into evolutionary algorithms, and we can test various features. So you've got the coding idiotypes on the left-hand side. Then you can see that we can vary different things like tolerance to drought, rooting efficiency, etc., and run through this algorithm until we're not getting any further improvements. And when we've got to this stage, we can look at the performance of our future crops with different designs and see how they perform reliably in different environments. So this just shows the result for both our current cultivars and our newly designed cultivars and what we expect to be able to achieve. So what I would like to emphasise is that this is work which is done in collaboration with many people around the world. Although we're very egotistical at Rothamsted, we know that we can't do everything on our own, and we're keen to collaborate. And we're building strategic links and scientific collaborations with other key players, both within the UK and internationally. So I guess this is where we want to be. We would like to get some wheat ears of this size, in fact, a whole field full of them, and actually make sure that we meet that target. So thank you very much for your attention. And if you've got any questions, I'd like to answer them.
Anyone have any questions? I have one question, I guess.
Yeah, I can still hear you.
Can you hear me, Martin?
Yep.
My only question is your main part of your research on yield improvement or on efficiencies of your wheat? Why 20 tons? I guess that's my main question.
We believe that we're going to need to increase yields to meet the demand. So therefore, we believe it's important to increase the potential. We know that there is a strong relationship between the yield potential and the actual farmer's yields, even though they might be, on average, significantly lower. But if we increase yield potential, we'll increase them generally in the stressed environments as well. But in doing and pushing for increased yield, we also know that we need to increase the efficiency with which we're using resources.
Yeah, because
I mean, both things are part of the program, but the key thing that our key target is to increase yield.
How do you see that affecting the quality of your wheat?
Okay, so there is going to be a strong interaction between the two, and that's why it has to be an iterative process. if we simply double the amount of yield and we don't get any more nitrogen into it, then the quality in terms of the protein content is inevitably going to be lower. So we have to look at ways of also increasing the nitrogen content, perhaps by looking at better remobilization, and perhaps by looking at other sources of nitrogen fixation for the crop. So we're not necessarily just having to pour more nitrogen on it to sustain those high yields.
Yeah. Anyone else have? Okay, yeah. That is my one question, or that is my one concern. Is that, yeah, 20 ton, like doubling my yield is great, but if all my neighbors double their yield and my price goes in half, I'm no further ahead as a farmer, unless I can cut my nitrogen maybe in half. You know what I mean? Like to me, I don't need to feed the world. I need to feed my family, right? So that is, I guess, if I don't produce double, the 9 billion people or whatever the target is not going to be there. If we don't produce it, the people won't be there, right? They have nothing to eat. They won't reproduce. I'm sorry, that's just a hard truth, right? I'm not trying to. I'm sorry. I'm not trying to. I think you're doing great work, and I mean, if we can do more with less, that's all. That's what I really would like to see. But sorry, I'm stepping on you trying to answer my questions.
I guess what we're trying to provide is society with choices, so that we can decide not only how much we produce, but also how we produce the land. I know that in Canada you have quite large fields, but in much of Europe, for example, we don't have a lot of space. So there is a choice between having more land that we can use for other purposes, diversing, for example, into crops which we're using for bioenergy, because the wheat fields that we have are producing at a higher level. So a lot of what we're trying to do is offer society those choices for the future.
Yeah, no, that is. We had this yesterday with Ross's talk, right? We talked about losing land and land being used for other purposes, right? So I can understand that. So that's a very good point. Okay, sorry, we do have another question from somebody else other than myself, so we should let him talk.
Okay, thank you. I just would like you to comment on the fact that you're using winter wheat as your platform, and could you make some comments that might occur to you to compare winter wheat with spring wheat, and especially in Canada, we've got a short growing season, so our spring wheat varieties must look like a racehorse compared to a winter wheat variety. You've got a 2020 program for winter wheat. What might you think could be a goal for spring wheat?
I've never thought of a particular number for spring wheat, but I think you make a very good point, because if you look at the resource use efficiency of your spring wheats, they're every bit as good as our winter wheats. It's just they have a shorter period in which they've got to do everything. Most of the traits that we're looking at are generic, and if we improve photosynthesis, for example, then that would apply to a spring wheat just as much as to a winter wheat. But what I would hesitate to do is to suggest that one size will always fit for all environments. I think a lot of the traits are common, but if we actually want to design those idiotypes for the future, then it might be what works really well for me with a winter wheat crop in the UK, and the design that we'd come up with for a plant growing in Canada might be quite different. But again, it might be different again if we were looking at Mexico.
Could I follow up? I think you mentioned earlier on that your breeding program is supported mostly by public institutions in Europe, and in North America we seem to be reducing our public investment in research and plant breeding, and the focus is shifting over to the multinational chemical and genetic companies.
Yeah, so it's very much the same in the UK, that we used to have a plant breeding institute that was sold by the government in the 1980s, and has been replaced by commercial industrial input rather than a public sector breeding. We do have public sector research, and we have got a collaborative project called WISP, which is a joint project between the academics and those industry partners. So for the wheat breeders in the UK, they are quite small. They've got quite a small market, whereas some of the big biotech and chemical companies clearly have much bigger markets to aim at.
We'll have one more question, and then we'll finish up with you here, Martin.
Okay.
Yeah, Martin.
Dr. Perry, thanks for your presentation. I heard, or actually saw an article this past summer about some work in, I believe it was in England, about nitrogen fixation associated with some of our non-legume crops. And I wonder if you just comment on that work and whether you're looking at that as part of your platform here as well for nitrogen efficiency.
That work was being done at John Innes Institute, and they're trying to really recreate nitrogen fixation in crops which don't have it already. My only caveat with it is that it requires light energy, and if you're using light energy to fix nitrogen, then you're not using it to fix carbon, so you can't do both. And I'm not sure if that's necessarily always the best route, although I think it's good to have the research done to be able to work out then, again, for the future, is what combination of traits we want to put into our plants.
Okay, thank you very much, Dr. Perry.
Thank you, and apologies that I couldn't be with you today, and I hope the rest of the meeting's a great success.